Next presentation is it's a great time to talk to these guys. I want to bring David A. Dodd on now, Chairman and CEO of GeoVax. David, good morning. I imagine it's been a pretty eventful year plus 14 months for you guys, right?
Very much so. Very much.
Yeah. I would imagine that. So go ahead. Let's bring up your presentation. You can tell us all about the vaccines for unmet for unmet diseases that you're working on right now, because it's as good a time as ever, probably.
Sure. Thank you, Spencer. Appreciate it, and I appreciate everyone's interest in GeoVax. So I look forward to updating you. So GeoVax is a clinical stage company. We're well capitalized. We have recently financed the company and listed on NASDAQ. We have sufficient capital to take us through 2023 and be able to move our two lead programs into clinical development within the next 12 to 15 months. And I look forward to telling you about specifics of the company as we go through this presentation. We're focused on several areas of major medical needs, but also resulting in significant commercial opportunities. Our primary focus, however, is on advancing our universal coronavirus program forward into the clinical development, and I'll spend some time speaking about that and why we don't simply refer to it as a COVID-19 vaccine, but it's a coronavirus vaccine program, and our immuno-oncology. So those are our two areas of focus. We have other programs specifically in the hemorrhagic fever area, which are the viruses such as Ebola, Lassa, Marburg, and Sudan. They're at the top of the list of the federal government's bioterrorism stockpile program. Those are proceeding into clinical development, and we anticipate with continued success they'll become part of the government stockpile program, but they are being handled for us through non-dilutive funding by various arms of the federal government, which I'll discuss during this presentation. Let me take a moment, not to try and be a scientist, but to just highlight that we, as everyone, have a particular platform that we utilize in addressing both vaccines and our cancer immunotherapies. We use the same approach across all vaccine areas as well as cancer immunotherapies. And it's referred to as MVA, which stands for Modified Vaccinia Ankara, and then VLP is virus-like particles. Now, MVA was originally developed in the 1960s as a specific vaccine against smallpox to be used by individuals who had compromised immune systems. They either had comorbidities, they were elderly, there were just certain reasons they could not take the existing smallpox vaccine. It was too stringent for them. So this vaccine was developed. It's been tested in over 150,000 people. There are approved products on the market using MVA. So it's well recognized and validated and accepted by regulatory authorities. But at NIH, following the development of MVA, there was a team there that then repurposed it to become a carrier for other viruses, vaccines. And so that's what it's utilized and known for today. Now, some of the benefits it has is it is exquisitely safe. Not only has it been tested a lot, but it does not replicate in mammalian cells. It replicates very well in avian, which means chicken, duck type cell lines. So you can produce a lot of it using cell lines that are from chickens or ducks and all, but it carries no risk of infection to humans. So that's very important to it. It also gives you a potential single dose protection, and we'll talk about how we've been able to further validate that. And it's self-adjuvanting, which is nice because with the VLP generation, and the virus-like particles are particles that are generated as a result of how we utilize and encode certain viral proteins into the MVA. They generate particles that essentially are identical to those of the target pathogen, but they carry no infectious risk. So it generates a broad immune response, both T cells and antibodies, gives you strong durability, typically do not need boosters, do not need adjuvants. And as I mentioned, it is well validated and accepted by regulatory authorities. This is a cartoon that shows you how we utilize MVA. So MVA, as I mentioned, is a virus, but it's a large virus. and it allows for a considerable amount of antigenic protein encoding, so to speak. So if we were talking about, for instance, COVID-19, we're all familiar and have probably heard that the mRNA from Moderna and others, or even the adenovirus vaccine from a J&J or AstraZeneca, they all utilize what is referred to as the S protein or the spike protein. And that's the predominant protein, and that makes sense. And that would be depicted in this cartoon on this blue antigen. However, those technologies allow for only one protein element to be encoded into a vaccine carrier. MVA allows us to do multiple ones. In this example, we have 3. We could have 5. So in this example, the blue would be the spike protein, and we have other structural proteins that we encode, and that is, in fact, the approach we've taken. The importance of that is that you generate not only the antibody responses, but also the T cell responses, which is what's important for not only durability of a vaccine, but perhaps most importantly, memory is driven by that. So a lot of people are asking, what is the memory of these new technologies, mRNA or either adenovirus? And they say, are we going to have to go back and have boosters? And even the companies are saying yes. And they're pointing out they have to reconfigure probably as the variants emerge, because it's like annual flu season. You have to redo it every year. That's not the case, we believe, with MVA, and we'll talk about that. So the latest news that we continue to hear is coming out of many different parties, and as CDC has recently pointed out, we're seeing these variants emerge. So out of the United Kingdom, South Africa, and out of Brazil, now out of India. So we're seeing these different variants emerge, and it's a really emerging, and no pun intended, but a new threat related to coronaviruses, not so much just COVID-19, but we're talking about now COVID-20, COVID-21, or COVID-UK, or South Africa. And that has driven the approach that the GeoVax has taken. So we've constructed several vaccine candidates. They're in animal testing now. And they utilize not only the spike protein, but these other structural proteins. And the purpose and the reason we've done that is to be able to encompass what may be an emerging variant before it actually emerges, and thereby not have to do annual reconfigurations, as we're going to see is necessary with mRNA and adenoviruses. Now, on this slide, it states funding opportunities and identifies these 2 organizations, one of which is NIH. Both NIH as well as the organization known as CEPI have both recently issued calls for proposals specific to technologies that can address pan-coronavirus or broad-based coronaviruses in a single dose and cover more than just a single protein at one time. We're in very close contact and discussions with NIH and with BARDA and others because of that. We received our initial funding from NIH in January that we announced, and that enabled us to go forward because we're testing right now against the South African variant as well as other elements in it. Now, what we've been able to demonstrate historically in other models, we showed with Ebola through non-human primates. So we're ready to move into the clinic with our Zaire Ebola vaccine. We showed 100% protection in a single dose, no adjuvants necessary, and no boosters, obviously. And ours has been the only Ebola vaccine, including the Merck, which is approved now for human use, and the J&J, which is approved in Europe. But both of those required multiple doses in their non-human primate studies, and we've been able to demonstrate it in a single dose. We've also, in other animal models, with the Lassa, the Sudan Ebola, and the Marburg, we've been able to demonstrate 100% protection, single dose. And I'll touch on how those have advanced beyond the data here where we were guinea pig and mice, but they've continued to go forward into non-human primate. Zika is another example, 100% protection, single dose. Zika remains a major medical need and a threat in the southern hemisphere, along with other flaviviruses, which means dengue, chikungunya, etc. We've shown excellent results, and we're ready to move into the clinic there. We're in discussions with potential collaborators in the southern hemisphere to be able to move this forward, which is our strategy, is to move it with a partnership this into the clinical development. We think this will be a very important product. We have several near-term data milestones coming up. As I mentioned, with our coronavirus program, We're doing animal testing. Those results are coming in to us as we speak. So we expect to see those coming in yet this half of the first half of this year, as well as some into the second half. I mentioned previously the loss of Marburg and Sudan. Well, based on the results we've previously shown, the U.S. Army is now fully funding our Lassa vaccine through non-human primate in preparation of GMP or CGMP material to move it into the clinic. Those results will be coming in the second half of this year, and the Marburg and Sudan vaccines are also similarly being tested in non-human primates at no cost to GeoVax or our shareholders. And those results we're expecting yet this half, and those are being managed for us through NIH. So we've got some exciting near-term data milestones that will take us to the brink of clinical development. And based upon the discussions we've been having with BARDA, we anticipate that if the results for Lassa, Marburg, Sudan are anywhere close to what we saw with Ebola, these will continue to go forward with the support of these government entities. Malaria is a major need. Over half of the world's population is at risk of malaria. There's one vaccine. It requires 4 doses, has an efficacy rate of 30 to 40%. We've been working on utilizing our technology to be able to encode components of the virus at different phases of the virus, which we think will give a broader coverage, better prevention. And those we're working right now with Johns Hopkins University in their tropical medicine area. We've got results that should be coming in yet this year also in that area. So looking forward for the remainder of 2021, we have some very exciting data coming forward that we think will further stimulate the value of the company and provide greater financing. Many people ask us about the priority review voucher program, and I'll just simply say that it is a very important program. It's on our target list, but there are 6 indications we're working that if the products are successful, we'll be seeking such vouchers. HIV, we've had a long history in HIV vaccines. We continue to move forward, albeit extremely slow, because it's all based upon funding from NIH, and frankly, NIH has not demonstrated the level of appetite to fund and move forward into pivotal trials with HIV, certainly as they have in other areas of infectious diseases. And right now, this program is still going, but it's slowly, but our focus is on our other areas of coronaviruses and then our cancer immunotherapy. And related to cancer immunotherapy, our approach has been to recognize that one will continue with standard of care. And we believe we can enhance the performance of the immune checkpoint inhibitors, and that'll be by constructing a vaccine with our MVA VLP technology tied in with a tumor-associated antigen. Our first target has been the MOC1 tumor-associated antigen. This cartoon demonstrates it's associated with many different solid tumors, so it's a good target to have. And we have been working with Dr. Oya Finn, who is a world-renowned expert on this tumor-associated antigen at University of Pittsburgh. We've done, these are humanized mouse models against human tumors in a therapeutic experiment. So all the mice had the tumor. One cohort received no treatment. The lowest line there, the bottom line that you see at day 30, the growth of the tumor arrested and began to decline. That's the combination of a MVA-VL1 vaccine from us in combination with a checkpoint inhibitor. So that was a difference of almost 60% there. So we believe it has a very important utility there. And then we did a prevention experiment, meaning that they were tumor-free. One cohort received nothing. The other one received our MVA MOC1 VLP with a peptide, not even a checkpoint inhibitor. And it showed 100% protection against tumor growth versus 100% growth when a tumor was introduced with the cohort that was untreated. We use this as a model for recurrence prevention, not as true prevention, but for cancer recurrence. So someone's gone through treatment and you want to prevent and minimize recurrence. So we've been encouraged by these, and we're anticipating moving forward and being into the clinic next year, but we're putting everything in place right now, including the manufacture of the bulk drug substance, which is probably the biggest challenge that one has, but we're going forward with that, as you can see, and that's the timeline. So we're excited about that, while we're also proceeding forward related to our coronavirus program towards clinical development. We have strong intellectual property behind all that we do. It continues to increase and grow, and I'll just end by reiterating that our current focus is behind our universal coronavirus program, which we hope to have the results that will enable us to, in partnership with NIH and BARDA, be able to accelerate into the clinical program and move forward there. And then with our immuno-oncology to move that into the clinic over the next 12 to 15 months. And we do anticipate that upon success coming out of our current hemorrhagic fever vaccine programs that are in testing through non-human primate, that will receive the support, and those will advance into human clinical trials. And as I mentioned, we've already initiated discussions. They were actually initiated by BARDA towards us about the government stockpile program. So with that, again, I thank everyone for your potential interest in GeoVax, your time and attention, and I'm happy to take whatever questions anyone may have. Thank you.
Yeah, I do have a very, very in-depth question, very specific question from someone in chat right here. This person has done their homework. I'm just going to read it directly. RE, the strategic stockpile program, have you engaged in discussions with the government, the federal government, that could lead to a continued supply contract similar to the one given to Emergent Bio?
Yeah, that's one of the answer is yes. As I mentioned there at the end, recently the federal government reached out to us, knowing the data obviously that's underway, because the Army's handling the Lhasa, and then the Marburg and Sudan are being handled by NIH. So they initiated discussions with us about participation in the government stockpile program. We're aware of the value. If you look at the last product that went into it, it was a $2 billion deal over 10 years with the initial, so that's $200 million a year in value. You have no marketing costs, and the initial payment was over $170 million. So the answer is yes, we are in close contact on that. We do believe, obviously, we need to see the results that will come out of it, but keep in mind, Ebola, we've already shown 100% protection, single dose, et cetera, and we're hoping to see similar type of results with the other three, and that would enable us to be providing to the stockpile program, sort of the full package of these hemorrhagic fevers. So the answer is yes, we have engaged with and we are familiar, and we would anticipate something similar to that program.
And then I know you had the slide up, but can you just reiterate the timeline for any upcoming data
for the immuno-oncology?
Yeah.
Okay. So, excuse me. The timelines there, as we've shown, are that we're expecting to complete what we're working with now, what we call our IND enabling or preclinical verification, but we're about to start the manufacturing of the bulk drug substance. We're expecting that to start in third quarter of this year, and we would file the IND in early next year. And upon approval of that, we've put on our timeline sometime in third quarter, but obviously we'll be driven somewhat by how our data results come forward, but mostly because the preparation of the drug, the element to go in there, and then the IND approval. But that's what we're looking at right now is a third quarter.
David Dowd is the chairman and CEO of GeoVax. The URL is up on the screen. The ticker is up on the screen. David, thanks a lot. Appreciate it.
Thank you. Have a great day, everyone.